Hello and welcome. My name is Derrick Rossi. I'm the interim CEO of NYSCF, and I am excited to kick off our webinar today. I'm sorry I'm not actually able to be there in person. I'm traveling and I'm on the wrong time zone, but I did want to pop on with this brief recording because I'm really excited about the topic that we're going to be hearing about today. It focuses around a study that we recently published in Nature Neuroscience a few weeks ago, focused around post-traumatic stress disorder. So you will hear today about a study led by scientists from Mount Sinai, the Veterans Affairs Office, and Yale in collaboration with our team at NYSCF, where they focused on post-traumatic stress disorder, PTSD, and they used the stem cell-based model to study this to find some really important insights, suggesting that there's a predisposition to PTSD that is discernible and tractable even without knowing who had PTSD and who didn't. So what's important about this study is that it's really the first of its kind, using stem cells to address this really intractable problem. And it really illustrates the power of stem cells and what NYSCF brings to bear by using stem cell models from many dozens of donors, some all from the military, some of whom had seen or experienced post-traumatic stress disorder and others that hadn't, and using this model to uncover some of the really key insights or key insights that were hitherto unknown about this disease. So we're going to hear more about that today. So I'll turn it over to the team now to carry on the rest of the webinar.
Thank you so much, everybody, for joining us. My name is Raeka Aiyar. I'm the NYSCF Vice President of Scientific Outreach and Diversity, Equity, Inclusion, and Belonging. I'm very excited to be here today moderating this discussion. It's a real honor and a pleasure to have this group with us to take a deeper dive into post-traumatic stress disorder and how it affects the brain. It's been a really very challenging disease to treat, so this is a major unmet need that NYSCF is really happy to be taken on. And so we're really lucky to have this amazing group of panelists with us today to delve into their latest study on PTSD, which was published on October 20th in Nature Neuroscience, as Derrick mentioned. We're really honored to be part of this great team that created the first stem cell model of this disease. I know that our late founding CEO, Susan Solomon, was especially proud of this feat. So we're happy to be talking about this study and its implications for understanding risk for the disease. Only a minority of trauma survivors develop PTSD, and that's a really important challenge to address. Understanding how the disease affects our brains and the cells within our brains. And what treatment opportunities are now on the horizon as a result of this work. So I want to thank all of you in the audience who submitted questions in advance. We'll be weaving those in as best we can throughout the discussion. Please also add your questions to the Q&A function in Zoom as we go, and we'll do our best to address as many of those as we possibly can. It's now my pleasure to welcome our panelists. I'll start with Rachel Yehuda, an endowed professor of psychiatry and neuroscience of trauma. She's also director of mental health at the James J. Peters Veterans Affairs Medical Center. Our second panelist is Kristin Brennand, who is the Elizabeth Mears and House Jameson Professor of Psychiatry at the Yale University School of Medicine. She's also a NYSCF Robertson Stem Cell Investigator alumna. And last but certainly not least, Daniel Paull, our Senior Vice President of Discovery and Platform Development here at the NYSCF Research Institute. So thank you all for being with us today, and let's get right into it. Rachel, I'd like to start with you, and as the originator of this collaboration and study, you've been studying PTSD and the biological impact of psychological trauma for many years, with hundreds and hundreds of publications on it. we all think that we know what PTSD is, but I would love to hear in your words, what this disorder is, and to hear how your lab has been taking this on.
Well, thank you very much. It's great to be here. So PTSD is actually considered a psychiatric condition, but it explains a set of symptoms or phenomena that occur to people after they've been exposed to extreme trauma. If you don't know what it's like, it's kind of like being haunted by the traumatic memory of the thing that happened to you. And some of the symptoms of PTSD revolve around having intrusive, distressing recollections of the event, having nightmares about it, restricting your life so that you don't really have to think about the event, of being very avoidant and hypervigilance, kind of being on edge or startled because you fear maybe that the event will happen again. And it can happen to people who are exposed to violence or combat or first responders or natural disaster. So it's kind of like a stress reaction that would be perfectly expected in the immediate aftermath of an event, but it just doesn't seem to go away. It just sticks around for weeks or months, years, and sometimes even decades.
Yeah, certainly a really debilitating disorder. So can you talk about, you led the study that inspired today's webinar. Can you tell us how this came to be and how it fits in with the other work that you've been doing on the disorder?
Yeah, so I've been here at Mount Sinai for since 1991, which is a long time. And even before that, my postdoctoral work focused on kind of the biology of PTSD. We had an observation really a very long time ago in the late 1980s that people with PTSD had lower cortisol levels than people that didn't have PTSD. And at the time, this was kind of an inconvenient truth because the whole idea of PTSD was really controversial and people didn't quite understand it. And stress theory basically said that with time, all the biologic responses that you mobilize in response to stress kind of recalibrate. So the idea of finding low cortisol was almost a negation of the idea that people really continue to be marked by a traumatic experience and feel transformed by it. But the low cortisol,
could you tell us what cortisol is for those?
Yes, of course. Cortisol is the stress hormone that is released during fight or flight. And cortisol, along with adrenaline, are what really helps you do the kind of biologic response you need to combat the immediate threat of the situation. Usually it's to run away or to fight or something like that. Some people freeze. But the traditional fight or flight response is really initiated by the brain to release these stress hormones that help you do what you need to do in the short run. We noticed that cortisol levels were lower, but adrenaline levels were higher. So that kind of started us out on a very long path to try to understand what really happens in people that develop PTSD. And it became known to us very early on that, as you mentioned, only some people who are exposed to trauma develop PTSD. And we kept wondering if at the heart of the problem might be this idea that cortisol levels were lower. And we found in studying people very close to the event, or even people who were at risk for developing PTSD, that they had low cortisol levels too. So it became an issue of really trying to see who develops PTSD and if we could use some of the biologic information around cortisol to teach us. Now, over the years, people started to develop very sophisticated biomarker approaches, mostly to use in blood. And the question of risk for any psychiatric or mental health condition, people started to really think about genetic risk. Like maybe the reason that you get PTSD might be the same reason that you get depression or schizophrenia or other conditions like that. Maybe you have vulnerability genes. So big efforts started to occur around identifying genes through genome-wide association studies. But even if you do these, what they're called GWAS studies, you can identify potentially risk genes that might be associated with PTSD, but you wouldn't necessarily know how that gene functions in the brain just by being able to develop, just by being able to measure it in the blood. So the idea became, as people were starting to think about genetics, could we actually, if we found these genes, be able to determine whether they were part of what is going on in PTSD? And I guess the third thing that we were discovering was that when you gave stress hormones like cortisol to blood cells that were live, or even if you gave cortisol to people, they responded to these stress hormones in a different way than people who didn't have PTSD. So when I first met Dr. Brennand and she told me about the ability to use this kind of technology to kind of take cells back to their embryonic stage, I thought, wow, talk about doing a before study. That would be kind of amazing to see what the neuronal architecture or genetic architecture might be like very, very, very early on. But because PTSD requires an environmental exposure, that's where we got the idea to incubate the neurons with stress hormones, cortisol, hydrocortisone, to be exact. And that's when we decided that we might have a simulation in the dish of PTSD risk factor plus environment, and could we then from there find a unique gene expression signature that could distinguish people with and without PTSD. So when we first conceived of it, it seemed, wow, this is almost a science fiction idea. How are we really going to get it to happen? But we teamed up together, the three of us in our groups, and made it happen from there.
Fantastic. And we're going to get into what we mean by those gene expression signatures as we go along. But thank you so much for setting the stage there, Rachel. I think, Kristin, this is now a great time to bring you on and talk a little bit more about genetics, as this is your area of expertise. Things like twin studies and other genome-wide association studies that Rachel mentioned have told us that there is a genetic component to PTSD. So can you tell us a little bit more about what is known about the genetic basis of it and why it's been a challenge to unravel prior to applying stem cell models to this question?
Yeah, well, so we know that in general, sorry, I'm getting over a cough. In general, psychiatric disorders are highly heritable. At the high end, we have schizophrenia, where twin studies estimate the heritability to be over 80%. So that means while, yes, there's an environmental component, 80% of your risk for schizophrenia is risk that you're born with. Likewise, autism spectrum disorder, ADHD, and bipolar disorder are all also over 75% heritable. At the other end of the spectrum, we have PTSD and depression, where twin studies estimate the heritability to be more like 30 to 35%. And this also makes a lot of sense because we know that things like environmental stress is, an important part of depression risk, and that trauma exposure is a required element of a PTSD exposure. And so we have this spectrum of disorders from very, very heritable to very environmentally impacted, right? You could have the highest risk in the world for PTSD, but if you live a charmed life and you're never exposed to trauma, there would be no clinical impact of that risk. And so when Rachel came to me to ask if we could do a stem cell model of PTSD, I think I said no the first time. I said it was impossible. I said this was the tool that I felt confident applying to schizophrenia or autism, but that the heritability of PTSD was much too low, and that I didn't think we would see anything but in a baseline difference between PTSD and control-derived neurons. And that's when she was like, but that's not what I want to do, Kristin. I don't want to compare them at baseline. Of course, they're going to be the same at baseline. I want to compare their response to stress. And that's really when she got my attention. I was like, well, I love experiments where you're going to learn something interesting and important either way, because if I was right and there was no baseline difference between PTSD and control neurons, and if Rachel was right that they responded differently to stress, we were going to learn a ton. But even in the third possibility, where all neurons responded to stress the same way, we were going to learn a lot about stress response. And it just meant that the PTSD component didn't arise in the brain response. It might have been about how the blood responds to stress and what they're sending up to the brain, right? And so like it would have meant that hydrocortisol was the wrong stressor, or the wrong, like, or that PTSD patients would have a higher dose in the brain, or like that we were missing some piece. But we were going to learn something very interesting and important if the neurons all responded to stress in the same way when dosed with the same stressor at the same dose. And we were going to learn something very important if they didn't respond the same way. And so at that point, this was just a very, very ambitious, very challenging project that suddenly seemed like one we had to do, because we were going to learn something fundamental about the way that neurons respond to stress on top of everything else that was going on. And so then it became, how do we undertake the most ambitious project I've ever dared to dream about? It was audaciously bold. And that's when you guys came in. It was like, hey, I know you've been developing these robots. I've got a really, really clever experiment that will require all of your robots, and an amazing cohort that's already existing thanks to Rachel's efforts.
Thank you. Well, that's actually a perfect segue then to Dan, as to how NYSCF came to be involved in this study. Kristin talked about the challenges with developing a stem cell model of PTSD specifically, just because of the stress response that's involved. And what's also at play, I guess, is the differences between all of these patients. Every patient sort of experiences it quite differently. That's pretty common with psychiatric disorders. So Dan, one of the developments that we're most proud of at NYSCF are these robots, are the automation that Kristin described, that you've played a central role in developing. Can you tell us about why automation was so useful for this particular study and for taking on this unique challenge posed by modeling PTSD?
Yeah. So I think nothing epitomizes high-risk experiments more than this. I think people have already touched on that, and that's sort of been NYSCF has championed these high-risk experiments, and for us personally, one of the big ones was, can we use automation and robotics to be able to perform experiments with a level of reproducibility and control that just technically by hand is very hard to do, and then on top of that, scale. And so by scale, I mean be able to analyze lots of different people at the same time, because these diseases, like these neuropsychiatric diseases, like many different diseases, the way to uncover whether there is something wrong inside the cells or whether they can be a disease signature can be detected is that you need many people to sort of overcome any noise that might be there. And so using robots, it makes things a whole lot easier. So about a decade ago—
Can you explain what you mean by noise, actually?
Yeah. So noise in a sense is if you only study cells from 3 people or 5 people, because the disease might only manifest in a very subtle way, it's sometimes hard to detect that if you don't have enough samples. You can't quite be sure whether what you're seeing is true or not. And so the only way to do that is to just keep increasing the size of the study. And again, doing that by hand, I've done this for many years, is just a lot of work and can be challenging in that way.
Right,
so,
to pick out what's specific to the disease versus what's just related to differences between these individuals.
Precisely.
Yeah,
precisely. So about a decade ago, a little bit over now, we started bringing these robots into the lab with a very sort of ambitious goal of being able to, at that point, simply make stem cells from lots and lots of people in parallel. And so we were able to get that up and running in really, really impressive way. And so that led us to sort of being an ideal collaborator in this study, because not only could we help make the stem cell lines, and I'll come on to that in a second, but we could then also use the robot to turn these into neurons and do all the rest of the things we did in this study. So
I believe there's
at
a
the heart of
video
it-
we can show of how
Yeah.
these robots are working while you describe
Perfect.
this.
So at the heart of it, these robots do everything that I would do by hand as a scientist in the lab. You can see these plates being moved around in this video. On top of them is sort of a pinkish solution. That is the food that these cells eat. And our robots run 24 hours a day, 7 days a week. When we're at home sleeping, they're taking pictures of the cells overnight, but otherwise doing everything that we would do as scientists, we've been able to recapitulate onto the robots. And so for us, our involvement in this study began right at the very beginning, where thanks to Rachel and her team, we were able to collect samples from veterans who had served overseas, and half of which had returned with symptoms of PTSD and clinically diagnosed PTSD, and half hadn't. And so we were able to take blood samples and skin biopsies from these individuals and put those straight into our robots so that we could start turning them into stem cells. And so for anyone that's not familiar, the technology that we're talking about here is called induced pluripotent stem cells. And so this is a technique that was discovered, again, a number of years ago now. But effectively, if we turn a few key genes on that are normally turned off in our cells, if we turn them back on, we can revert a skin cell back into a stem cell. And once it's a stem cell, you can then go anywhere you like. And so for us in this study, we've then taken those stem cells and turned them into brain cells, into neurons that we're able to then analyze. And so, again, you've sort of heard from it. It's a disease that maybe isn't an obvious one to go after, and so there hadn't really been a lot of people investigating this with stem cell models, and so it led us up to being in a really good position to go tackle it. But again, using these robotics allowed us to tackle it from the get-go with a very large, cohort of patients and donors, so that we could have some confidence in what we were seeing should we eventually see a result from this.
Fantastic, thank you. So let's move on to what you actually found in this study now that we understand, how this sort of unique combination of people and expertise came together. Dan, you talked about focusing on neurons. Rachel, you mentioned a lot of other studies have used blood samples, which you also did here as a comparator. Now, there are a lot of different types of neurons that are specialized for different functions. What specific types of neurons were used here, and why did you select those? Dan, do you want to start?
Yeah. So for sure, there are an ever-growing number of brain cells. It seems we keep the more sequencing we do and able to delve into the brain, the more we seem to unlock these different subtypes. But for us here, we chose one particular type of neuron. It's an excitatory neuron, again, in part due to the ease of making these cells, the reproducibility of them, and sort of our ability to handle them on the robotic platform, it's sort of an ideal starting point. But it's a starting point, and we can come on to that later. And so these cells have been, a major cell type in the brain, and one that we've sort of investigated before in other diseases. So it was a useful starting point for us in this study. We did investigate sort of different types at the very early onset of this study, but decided upon this one for again those reasons I just said, of reproducibility and sort of relevance and different things like that. So that was why we ended up with where we are. Again, a testament to the team that worked on this and the team that has built the robotics over the years, and in particular Tom Richter-Valich and Barry McCarthy and Jordan Goldberg and a dear friend of ours, Reed Otto, who passed away sadly. Their work was phenomenal in making this happen, and so getting us to this point of being able to make these neurons so reproducibly is why they were used and led to the results that we found.
If I could add to that just a little,
Please.
I mean, we actually spent a few years, if memory serves, really trying out different brain cell types to measure. And the consideration here was also to see which brain cells respond to the stress hormones, because they don't all respond equally. And this was a very important consideration for us. So we weren't choosing brain cells just because they were easier and reproducible. We chose the ones that were responding to the stress hormones, and they don't all respond alike. But in our follow-up work, we have now started to look at inhibitory neurons because we do need to have that very essential comparison. And when you get to the real true application of this work, which is to try to help figure out what drugs work in PTSD, these little distinctions between how different compounds will work on different brain cells become critical, and having the ability to do this robotically will be an extreme game changer for how we advance this technology in the service of the treatment of PTSD.
Definitely one of the most exciting implications of the study, and we'll talk about that. Rachel, I want to come back to something that you were talking about, which was the simulation of a stress response in these neurons. Can you tell us a little bit more about how exactly that was done, and what you learned then about how it is that our brain cells respond to stress or trauma, or as best as this simulation could tell us?
Yes. So before we embarked on this study, our lab had been working very intensively in the biomarker for PTSD space. We were very interested in not only learning about what the biology of PTSD is, but in trying to develop a blood test to diagnose PTSD. And one of the things that we learned after, I would say, more than a decade of studies, is that when you take a blood sample from people with and without PTSD, there aren't going to be all that many differences between people with and without PTSD in what I call static blood, or just dead blood cells. It's not like a pregnancy test. It's not like something that you can have or a COVID test, or something where you can look at something very, very clearly, but that there were teeny little signals that would come in and go. But once we, but in contrast, when we added stress hormones to live blood cells, all of a sudden we found a lot of things would pop, and that the differences between people with and without PTSD became very robust, not a baseline, but in response to just adding stress hormones to live blood cells. And that's the work that we did before going into the study, that actually I had to show to Kristin to get her excited about this, that people with PTSD, it's very deceptive. They don't look different than other people. It's not a wound you wear. And even at baseline, for a lot of biologic measures, they seem fine. Two people with PTSD can be walking down the street. You won't be able to tell them apart. But you know what? If a blast goes off, the person with PTSD might really duck and have a very exaggerated response to stress, whereas the person without PTSD might have a different response. So the biology and the behavior of PTSD comes out upon provocation. And so that was the blood work that then led us to say, well, you know what? Maybe from blood we can get biomarkers. We might be able to get a blood test. But what happens in the blood is not necessarily what happens in the brain. And one of the most important findings that we had in this study is that indeed the overlap between the genes that are expressed in blood and brain when you deliver stress hormones to live neurons and live blood cells is pretty small. And so that they have some in common, but they also have very different things that are activated because blood and brain are different.
Absolutely. A very important comparison to be able to do in this study. Dan, could you talk a little bit about what we learned about how these brain cells across all these different individuals then responded to this stressor?
Yeah, so Kristin and Rachel said, and sort of we touched on it, but wanting to have a cell type that would respond to a stimulant like cortisone was critical at the beginning. So one of the first things we confirmed is that yes, indeed, these cells that we had picked to study do express receptors that sort of meant that hopefully we would see a response. And sure enough, we tried various different doses of elevating the stress levels exposed to the cells. So they were sort of a more mild and then a highly elevated dose, and just saw an enormous change in the overall gene. So that's sort of, if you looked at everybody without stress, without sort of breaking apart PTSD or not, there was just an enormous change overall once these neurons were exposed to the stress hormone. But where then the study obviously became of critical interest and real importance was that as we looked at the genes, so when we say look at genes, what was, in all of our cells at any given point in time, the genes and the way our cells are behaving differ, whether they're a skin cell or an eye cell or a brain cell. And so we can measure these things, we can measure these changes using things you might have heard of, like RNA sequencing. And so that's the technique we used here to sort of really dive in and understand which genes are being turned on or turned off, and whether they were different between our individuals from different groups. And so sure enough, this image will never leave my brain the first time seeing it, of just this incredible ability for us to distinguish based on a signature, if you like, of the cells that came from PTSD patients and those that were the healthy controls in the study. And they just completely sort of correlated. There was a signature associated with one group that was different to the other. And this really was what we were after, and we didn't know if we would see this. And so being able to detect that has been quite phenomenal. These genes, again, upon being stressed, showed a number of interesting sort of results. They overlapped with studies in other neuropsychiatric disorders, which is quite interesting. There's a lot of overlap in sort of existing data sets that came from other studies, which is certainly one interesting avenue. We saw an enrichment of genes that were associated with areas of the genome that have been identified as being risk factors and risk areas for developing PTSD. So again, another key part of this study was finding that. And then things that we were actually able to detect. So again, for example, if you give a stimulant like cortisone to a neuron, how does it behave? And one of the early things we found is, well, it seems like it could, if you give it very, very early in a neuron's life, it might inhibit the way these neurons grow and whether they form synapses together. And so actually Kristin's lab was able to help validate this as well. And from that finding, we went back as part of sort of in the process of publishing this paper, is actually test that specific thing. If we gave the stimulant at the beginning of these cells' life, if you like, as soon as they went from a stem cell to a neuron, sure enough, we saw this inability for them to make neural networks in quite the same way as our controls that were untreated. And so this isn't just sort of hypothetical data that we've discovered here. It actually bore true in validation studies that we've done in the dish. So a whole heap of different results have come out of this. The big take-home one, obviously, being that we have this signature that allows us to distinguish the patients, the PTSD patients from the controls. But the data set is expansive, and I think is going to now be supplemented as we move into these other cell types that Rachel just mentioned, whether it's another very specific cell type like an inhibitory neuron, or whether it even begins to get into more complex cell types. What if we took other brain cells like an astrocyte and looked at the effects in there, or even much more complicated tissues that will be harder maybe to decipher, but things like there's these cells that we can make called brain organoids. And so these are an accumulation of different cells that you would find in the brain, and they form a small ball, if you like, of cells. And again, there you've got lots of interactions that are happening between the different cell types, and so there the response that we see may go on to be different again. And so this study has really sort of teed us up to begin exploring these results in these other cell types.
That's really exciting. And when you think about then how to translate what we're seeing in the dish into, how does that then impact the brains of these patients? I wanted to pick on one thing that you said about the stressor happening, early in the neuron's life and that damaging effect. Rachel, maybe you can comment on this. What does that tell us potentially about earlier exposures or childhood exposures to trauma versus something that maybe happens later in life?
Yes, it's an important question. And I think that almost everyone agrees now that earlier developmental trauma can have really intransigent, long-lasting effects and shape the way that the neuronal networks develop and therefore dictate how people will respond to stress in the future. It's very important because, you know, when I went to graduate school, which was really a long time ago, the prevailing model was that people can, you know, get over earlier assaults because of the plasticity that is involved. But actually, the plasticity that is involved allows you to really incorporate the environment even more so. Now, that doesn't mean that you're doomed, but it does mean that you're changed. And so the fact is that environmental exposures change us. And that is really one of the important things that we demonstrated. And that's what Kristin was saying before, that we were going to learn something interesting, even if it wasn't going to be about PTSD, because this question of how early do you start to see differences in response to an environmental provocation is really something that has not been well demonstrated in humans because we haven't been able to look in this kind of way. And so that's why even removed from PTSD, this was going to be kind of interesting, but from neurons that are just several months old, we can start seeing that indeed they respond and how to just a single kind of provocation of stress hormones and in a dose-dependent manner. We didn't just use one dose. We used a bunch of doses in order to be able to look at this.
A
I do want to
related
jump in and add one point real quick.
please, Kristin.
Raeka.
Yeah.
I think one of the things that I do want to stress that really enabled this study was how well characterized Rachel's cohort was, right? Because if we just took regular controls off the street, we wouldn't know what their trauma response was. And so every one of our controls was combat exposed. And so we know they all were exposed and either had PTSD or not. Whereas I think in a traditional control cohort, you wouldn't have that information. And more than that, Rachel knows these patients and she knows who had childhood exposures and who didn't. And so she was able to look and make sure that we weren't biasing our study because we had more childhood trauma in one group or the other, or more confounds from depression or other psychiatric disorders. And I think a lot of these issues could really wreak havoc on a study if you started thinking about them post hoc. And so I want to put credit where credit is due. And I'm pretty sure her team agonized over, of the hundreds of patients in the clinic, which 40 to recruit for the stem cell study. And how do we, again, and Daniel talked about noise, variation in neurons in the dish. And we know this happens between individuals and experiments. It's also noise amongst people. And so I want to say that what was really helpful here was having, like, we could go post hoc and look, what was the childhood trauma exposure score in our cohort and see if that was impacting our results.
Yeah, thank you. That's a really important point. And there's a related question that came in about, the idea of chronic trauma and recurring trauma, such as, abuse that happens over a lifetime. Can you comment on that, Rachel, perhaps, or any of you?
Yes, I mean, it is the rare person that just has one trauma and then we're done. And I think that this is something that makes PTSD and trauma so hard to study, because the fact of one exposure changes you so that the next exposure has a different impact than if it would have happened without that first exposure. And that's why we have not been able to really get too far in our current studies, such as they are, and translate animal studies, which are pristine, to the human condition, which is messier. But what's really, to add to what Kristin said, what's really nice about this cohort is that now we are starting to do research with psychedelic drugs, and we're drawing from this cohort of patients so that we have an opportunity to use the findings that we have collected to see whether we can predict responses to certain medications. And we are even able to take these same cells that we have generated and be able to test how they respond to any number of different kind of drugs. So when you have this kind of a stable cohort that is relying on you for their care, at some point you've tried so many things, they're still your patients. We might be able to, in the future, have a very elegant way of being able to identify before we start to do a whole lot of different treatment approaches, who might be best suited for what kind of treatment. And that's really why all of us got so excited about doing this study, because as Daniel said, it was kind of impossible to think about, and very, very ambitious. And since I didn't know very many of the technical details, I just kept saying, but we got to do it, because I didn't actually understand what I was asking at some times. But we all knew that the payoff, if we could do it, would be very, very large. And that's really why we're here today, to communicate it, because it's really important for the public to have hope that in the future, in the near future, we will be able to use these kinds of scientific advances in the service of their healthcare, in the service of being able to kind of have better strategies for deciding how we treat patients.
Absolutely. And let's bridge into, the sort of implications for treatments. But one thing I want to make sure we address is that you did see in these neurons a hypersensitivity in those who we knew had PTSD. So the PTSD sort of prone neurons seem to be hypersensitive to the stressor that was administered in the dish. Can you talk about that, what you sort of saw there, and how that might pave the way for finding new treatments? Dan, do you want to maybe take that one?
Yeah. So, interesting, actually, some of our biggest responses came when treated with the slightly lower dose as opposed to the higher dose, and that's in part maybe because we can rather than just swamping these cells and everybody, whether it was the control or whether it was the healthy individual or whether it was the PTSD patient, everything just gets swamped when you just give such a high dose. This more lower dose, to Rachel's point earlier, it's this sort of hypersensitivity that might actually be the reason why we were able to tease apart the effect at this lower dose. And so again, the effect that we saw for us was this change in how genes are turned on or off in these cells at different times. And the work now is to understand, okay, so what does that mean? That's for sure a question that deserves a lot of follow-up, and there's a large number of candidates to go after. Based on genetics and based on things that we've learned from previous work, there are some that are maybe at the top of our list of things to go after and go to look at. But where it begins to then tie in is, okay, so now that we know things that are turned on or turned off, can you modulate that? Can you change that effect? So in addition to giving a neuron a stimulant like cortisol or hydrocortisone in this case, what if you did that and then followed it up with a drug of some sort? Would that dampen the response? Would that change the response so that the signature that we were seeing in these patient samples would sort of get wiped out to some extent and make them appear much more in line with the healthy controls? I think that's sort of we can chat more about that over the next few minutes, but that's sort of, I think, where ultimately this study begins to go. That's the natural path for it to keep going down. But there's a lot of baseline neurobiology that we can learn from this as well. Just simply because, again, of the data sets that's been generated in this study, and how we'll be able to tie them together to work that others have done in the field as well.
Fantastic.
And I want to add how many extra we didn't stop at this study. And I want to highlight again just how this team's been pushing forward. And so this first study was 20 combat exposed controls in 19 cases. And Rachel has been determined they were all men, and Rachel's been determined to add women so we can begin to understand if and how PTSD risk differs across sexes, because we do know that even in the military, women experience different types of trauma than men. And so we want to make sure that the information we're learning benefits all people who experience PTSD and not just men who do. And second of all, we started with one major cell type in the brain, glutamatergic neurons, but we don't think that's the only one implicated in PTSD. And so we've already now expanded this to a second cell type, GABAergic neurons. And I don't think we're going to stop there. I think what's really going to be important is mixtures of the cell types implicated in PTSD and understanding the way these cell types act in lone and in circuits, and really getting to higher level links between the genetics and the activity differences that are happening in response to stress too.
Thank you so much for, yeah.
Sorry, Raeka, just to piggyback off of that,
please.
just to kind of reiterate, I guess, why this is now possible. You know, in the past, these studies couldn't be done. You could use a mouse and you could use an animal model, but that, you know, that's a very different model for all the reasons. But nor can you go into someone's brain unless it's post-mortem to look at the effects. And by that point, it's quite likely that, you know, the changes have happened, and it's something very final in a post-mortem brain. You can't go into someone's brain while they're alive and just pull out some neurons because you want to look at how their response is in real time. And so having, you know, again, this cohort that Rachel's collected, a patient cohort that Rachel has got to know and developed, we now have the iPSC cohort. So these stem cells sit in our freezer. We don't have to go all the way back to the beginning. In fact, what we can do now is supplement them and just keep adding more samples to this cohort. And so at any point in time, I can go into the freezer, take out some of these cells and grow more brain cells if we need to, because we want to test something new. And so whether it's a new cell type, like Kristin just said, whether it's because there's a new drug that we want to test, and I think we'll come on to that in a second, or just whether it's a new question to ask, this is now there and it exists. Rachel?
Yeah, just to make sure everyone knows that the people that participated, the veterans consented to that.
Yes.
So this is a situation where we told the veterans very clearly that one of the things we can do is store their samples for future use, and we had an explicit permission to do that. And again, the ethical issues involved in doing this work are very important. And so it is, since we collect the consents, that's our role. And we do have to explain to people what can and can't be done with their samples. But for all the samples that are now in your freezer, those are people that have consented to have investigators ask questions. And it's the gift that keeps on giving, really. And it would be important to be able to add to your cohort so that we can ask more powerful questions, including the sex differences. The reason that's important is because statistics show that women are more vulnerable to developing PTSD. Now, I've always thought it's because they're exposed to more trauma. But if there is a genetic reason that they're more vulnerable, this is the kind of study that will find it, because this will be able to, it's the difference between having a baseline difference or a difference in response to provocation that is simply a difference between men and women, or males and females, I should say, more technically.
Yeah, thank you for raising that equity issue, Rachel and Kristin. You know, certainly the issue of women being affected more often than men. And, you know, we've also seen racial disparities as well. There was a question that came in about that. Like, for example, Black Americans have a higher risk and lifetime prevalence. So, you know, can you talk a little bit about what opportunities we might have now with the stem cell models to be able to delve into these? And I also just want to say we're obviously very grateful to all of these participants in this study that have, you know, really allowed us to look at it in a whole new way. And, you know, that, and thank you for pointing out the, you know, kind of ethical issues around that, Rachel. Yeah.
It was a racially diverse sample. So in science, you can make a decision to kind of control for these factors and kind of push them out of the picture in terms of adding variance to the data, or you can decide to actually study the very individual differences. These are two completely different statistical approaches. We chose to control for them, I believe, in all of the analyses. because we don't have a tremendously large sample. But it would be a very important idea to be able to do some kind of sub-analyses or secondary analyses to see whether there are racial differences in the response, because I think Hispanics also have been noted as having risks a twofold higher risk for developing PTSD. So this is where these kind of questions become very important. These questions can be charged and loaded. And this is a nice way to be able to examine things from a time where hopefully when we were all in our mother's embryo at 6 months, this was before any of those disadvantages started rearing their heads, but maybe not. But it's a way for us to begin to look at that. So I think that's a great future direction for us.
Yeah, thank you for pointing that out. I want to come back to the idea of the genetics. Kristin, I'd love to get your take on this now that we understand a little bit more about the risk and have these gene signatures of different expression between these two groups. How do we now integrate this genetic knowledge into finding new strategies for treatment?
Yeah, I mean, I think one can imagine a few different ways. The first would be if we knew there were a subset of people who were at higher risk. One possibility would be to tell them, to discourage them from being frontline medical worker, first responders, to discourage them from being in the military. I don't know that that's a feasible direction. I think people want to have control over their choices, and that we've historically found that giving genetic information without having actionable treatments isn't always something that people want. Although I do think it would be interesting to consider if the military, given that there are lots of jobs in the military, if or should they consider risk in proportioning these jobs. And this is an ethical dilemma that I don't pretend to know the answer to, but it's something out there. I think more likely to be adopted by individuals and the military is this idea of knowing that if you are trauma exposed, we could intervene. And so, Rachel's been involved in studies where in the ambulance for rape victims, or maybe in the battlefield, we could put in a drug to rapidly inhibit hydrocortisol levels in the blood and bring them back down. And would a fast, and if that's a perfectly safe drug, you would give it to every trauma exposure. And if there's risks involved, maybe you would limit giving it to those people who you knew were higher risk. I think every drug, there's this balancing act between efficacy and side effects, right? We give folic acid to every pregnant woman, even though not all fetuses are at risk, because it's so safe. And so your prevention here, you could just give it to all military members for their entire careers if it's safe enough. It really is going to come down to what the long-term consequences of continually suppressing stress hormones are. And then the idea that I find most intriguing is, as we better understand the risk, and this is true across brain disorders, we're finding that there's points of convergence in individual risk genes, that some people might converge on these factors or somebody else here. And this moves us towards bucketing patients, right? I think the idea of personalized medicine, one drug that has to be uniquely discovered for each patient, that seems terrifyingly unscalable to do a drug screen and approve a drug for each person. But if we can begin to figure out that there are 20 or 50 actionable pharmaceutically responsive subsets of PTSD, if the genetics can say these people who converge on signaling on this pathway or in this cell type respond best to this drug, you could start there in an informed basis. Right now in psychiatry, we tend to just what are your symptoms? Okay, often this drug works best for that symptom, and if that doesn't work, we'll just try a new drug. And if that doesn't work, we'll try a new drug. But every drug has side effect profiles. No drug is perfectly efficacious. And if we could go in with some knowledge of the genetic risk underlying those symptoms, we do not treat every cancer patient the same anymore. We ask, are they BRCA1 positive? Are they estrogen receptor positive? And we might treat a breast cancer and ovarian cancer the same way if they share the same genetic markers, even though the symptoms are completely different. And I'd like us to get to a point maybe in psychiatry where we are letting these genetic markers inform treatment,
Yes.
because they might better predict treatment than symptoms do.
Yeah, really, really great point. Speaking of treatments, there have been several questions that have come in about psychedelic drugs, which are a new sort of frontier in being able to treat some of these psychiatric disorders. And Rachel, I know that you've been working in this area and are about to start some trials. Could you speak to that a little bit?
I can. First, I want to just say on Kristin's answer, we are indeed doing studies where we're trying to prevent PTSD by administering stress hormones in the emergency room, and that and I think that not having knowledge is better than not having knowledge. By the way, the genes that are implicated in PTSD risk are not necessarily genes that will damage you. They may be resilience-related genes. We don't know enough to understand what we want to do differently in people. So it's kind of a wait and see. But the idea of being prepared or knowing what your risks are may make you take better care of yourself. They make you do the right things when you are exposed to trauma. And so there's great value in having this information, just like when people know that they are at risk for cardiovascular disease, maybe they'll take better care of their diet or something like that. So knowledge is a good thing. Being treated early or being hyper aware early about potential impact of stress can also be important. And we're studying psychedelic assisted psychotherapy now for PTSD. We're studying MDMA and psilocybin, more in chronic cases. But I can imagine a future where these kind of treatments are done earlier on in the course of therapy. And so I think that is a very exciting horizon. And a lot of people are a little nervous about psychedelics. We don't exactly know how it works. But these are the kind of studies that can provide a lot of reassurance that there is a way to understand how these drugs work and to take the mystery out of it, maybe even the mystical out of it, by just looking at some of the biologic facts surrounding how these drugs may impact people. And again, our combat veteran trial with MDMA has started, and we are going to be able to compare real-time treatment with results with some of the results from people in this study. So I think that will be very interesting.
Thank you, Rachel. We've only got a couple of minutes left, so I'll just ask sort of a closing question about what each of you are kind of most excited about in terms of the possibilities that are now on the horizon for the future of treating or preventing or intervening in PTSD or similar psychiatric disorders. Dan, I'm going to start with you.
Yeah, so I think it's two things. One is again, thanks to NYSCF and things like this study, there's a platform here now, and the platform is, I'll come back to PTSD in a second, but the platform is agnostic in some ways. We can take large cohorts from other diseases. We can take cell types that are specific to a given disease and be able to investigate them in much the same way we have here. I think this is one whole area that's just getting very, very exciting generally. I think specifically coming out of this study, we've touched on other cell types. There's a lot more to delve into to understand maybe the biology of what we've seen here. But Rachel just said, we don't necessarily understand what some of these new therapies actually do in these cells. And so, yes, we used cortisol here because we wanted to invoke a response. But in much the same way, if we were to take psilocybin or, you name it, drug X, and put it onto these cells, maybe we can actually begin to understand mechanistically what is happening inside these cells in response to those drugs. So those are the two areas for me. One is just more biology and increasing our understanding of biology, and one is increasing our understanding of interventions and perhaps finding better interventions.
Right, absolutely. And finding, different ways to understand the effect of these hormones or these these potential treatment modalities directly on neurons. Kristin, I'll turn to you next. And, I know that you also do this research in a lot of different psychiatric disorders, as you mentioned. So would really love to hear your perspective. You're on mute.
Just this morning, I was chatting about these exact questions with my team, some of the follow-up from these studies. And so Karina, who led a lot of the analysis at the end of the study, one of the things that she wanted to understand was why the neurons from PTSD cases were hyper responsive to H-corp. So she was looking for drivers of this. What genes were responsible for this? So genes that either went up, we predicted the genes that went up when you're stressed might be the drivers, and genes whose targets went up would be even more likely to be the drivers, because if they went up and their target genes went up. So she focused on transcription factors and I actually highlighted a handful of them. The one that comes to mind was CMYC, that its expression went up faster in PTSD cases when stressed, and its target genes went up disproportionately in cases when stressed compared to controls. And so now what we're looking at, well, if that gene necessary and sufficient for this exaggerated stress response. So we're manipulating MYC levels genetically and pharmacologically to ask, can we tamp down that exaggerated stress response? Is this actually a target? And maybe it's a terrible target, but who in its network? If we change this pathway, we prevent the exaggerated stress response, maybe we can prevent PTSD.
Very exciting possibility, certainly. And Rachel will give you the final word. Would love to hear what excites you, and also, how this connects to the work that you've done on intergenerational trauma. There were questions that have come in about that I know you'd like to address. So please go ahead.
I'm excited about this team and the fact that they have such great questions that are going to solve things that we haven't been able to solve, like how to prevent PTSD and what are the drivers of these biologic differences. And for sure, we're going to do the study where we're going to expose cells to different psychedelics. I can't wait for that. So I'm just happy that we were able to start something that doesn't need to end, and that can give a lot of young scientists the opportunity to spend years, if not decades, just really going so much deeper, because now we can go so much deeper than it was ever possible to imagine when our work began on this in the early 90s. So I just have so much gratitude for the New York Stem Cell Foundation, for Kristin and her team, for other collaborators at Mount Sinai, for my own group. We talked about my cohort, but the clinical team that works here did all the work. We have the most excellent and compassionate PTSD providers that can actually get the trust of our combat veterans to participate in this kind of research. So I'm very, very grateful to everyone. And I should mention also the Department of Defense who funded this work.
Absolutely. Well, gratitude is certainly a great note to close this discussion on. So I want to thank all of our panelists for a really hopeful, exciting, and intriguing discussion, and all of you in the audience for your questions. I apologize if we didn't get to all of them. We'll try to address them in the write-up that we do following this. So please keep your eyes posted for that, your eyes peeled for that. Hope to see you all at a future NYSCF event. So please stay tuned to your emails and our social media channels for upcoming webinars and in-person events we hope soon as well. So take care, and wish you all a very healthy and happy holiday season. Thank you.